Hello, my name is Raul Chavarria, and I study the evolution of the highly compact tardigrade body plan. My research is primarily focused on the genes that pattern the secondarily simplified anterior-posterior axis of tardigrades, also known as the AP axis. This is the axis that runs from the head to the tail of bilaterians. In many animals, the canonical Wnt signaling pathway establishes the AP axis during early development. Later, this pathway interacts with the Notch signaling pathway to give rise to a mid-trunk region through a process called posterior growth. However, tardigrades lack posterior growth and the mid-trunk region that develops through this process while still retaining posterior identity. This suggests that tardigrades have a secondarily simplified AP axis. So, I am interested in determining whether Wnt signaling establishes the highly compact AP axis in tardigrades and understanding the roles of Notch signaling during establishment of the AP axis. I previously showed that although the Wnt pathway is present in tardigrades, they have lost many Wnt ligand coding genes. This time, I take a look at the Notch pathway and find that tardigrades retain the receptor component of this pathway, shown in yellow here. I also found that tardigrades have both DSL class ligands of the Notch signaling pathway. These are jagged/serrate, shown in green, and delta, shown in pink. I focus on 3 developmental stages, stage 11, stage 12, and stage 13, and utilizing in situ hybridization, I was able to use 6'3 as a genetic marker for anterior identity across these stages. At stage 11, the embryo is pretty much a tube within a tube, and during this stage, the embryo starts elongating along the forming AP axis, where it will end up taking its characteristic C shape by stage 12. Down here, I show the expression pattern of key genes during these 3 stages. Notch and delta genes are expressed in endomesodermal domains through stage 11 and stage 12. At stage 13, delta expression can be seen highly expressed in the head region and also in each trunk segment, possibly specifying central nervous system. Out of all the Wnt ligand coding genes in tardigrades, I found Wnt2, Wnt4, and Wnt16b expressed in a posterior domain during the earliest stage of AP axis establishment. The expression pattern for these Wnt genes varies at stage 12 and stage 13, except for Wnt16b, which remains posteriorly expressed throughout these stages. All in all, our data suggests that the compact body plan of tardigrades evolved by conservation of the AP axis establishment function of the canonical Wnt signaling pathway with loss of the later acting posterior growth functions of the canonical Wnt and Notch signaling pathways.